Hi, my name is Alexander Stoltz and I'm a senior. For my science fair project, I was studying the effects of lead toxins on the gene expression of bacteria. So, all over the world, scientists are studying the gene expression of bacteria because we are, in fact, going through an antibiotic-resistant bacteria crisis, where the bacteria is no longer being killed by the antibiotics. So, scientists all over the world have taken to studying gene expression of certain bacteria and seeing what affects it and how it affects it. And they're doing this by taking things and putting them in the environment with the bacteria, or taking things out of the environment and seeing how it responds to that, as a way to just try to better understand the bacteria. So the question being posed was, how does the introduction of lead in varying forms affect the gene expression and growth of E. coli? So for this experiment, I had 3 different forms of lead that I tried. I had lead flakes that kind of went in the bottom of the plate that I was making, and then whenever I would pour the agar, some of the lead flakes would kind of float to the top. I had lead pellets that would go in the bottom of the plate, and they would just sink and kind of stay down there. And then for the third one, I had 200 milliliters of water, and then I would put some lead pellets in there and kind of let it sit for 24 to 48 hours as a way to allow the toxins into the water, and then I would take that water and turn it into agar. So then my hypothesis was that if I add the lead in these forms to the agar of the plates, the gene expression and growth of the bacteria will decrease as the lead concentration increases. So a little bit of background is, like I said, we are going through an antibiotic-resistant bacteria phase crisis, and it's very bad, so people have taken to studying this, and I have always been interested in bacteria. I love working with it, and this has always just been a point of interest for me, so my goal was to attempt to mimic the experiments in a high school classroom. So I had to find a way to, one, see the gene expression of the bacteria, and 2, do something that would conform with the lab safety level 1 that is required. So a little bit of procedure for this is 3 days before I did my transformation, I made up the plates. I had ordered a Carolina kit so that I would be able to see my gene expression. I took a plasmid DNA from a bioluminescent jellyfish and kind of put that into my bacteria so I'd be able to see it. This picture down below actually shows the gene expression of the bacteria. It makes it glow like a greenish blue color. That's really clear to see, so I would be able to tell gene expression quite obviously. So then 3 days before the transformation, I made up my plates. I did all 3 of the lead types, and then I just kind of let them sit in the fridge. The day before the transformation, I grew an E. coli starter plate on just a plain agar plate. I ordered a Carolina kit with this, and came with a file of E. coli DNA. And I just took that and put it on a plate and let it sit in the incubator to grow for my starter strain. Then the next day, after 24 hours of growth on the starter plate, I followed the procedures in the transformation experiment to put the plasmid DNA in the bacteria. And then I took the solution that I got there and I put 75 mils on each plate and then spread it around and put it in the incubator to grow. After 24 hours of growth, I would take the plates out of the incubator, mark down how much growth they had, and then see if there was any gene expression or glow. And then I did the same thing after 48 hours. For this, I would see if there was any more growth and any more glow or any less glow, because over time the gene expression will become less and less strong because it takes so much energy from the cell. So some things I used in materials. I have, of course, had distilled water, lead pellets, lead flakes. I had ampicillin because one of the things that was also with this plasmid DNA was ampicillin resistance. So I would have one set of ampicillin plates and one set of just plain plates. And if the bacteria grew on the ampicillin plates, that made me know that I got the transformation and that this bacteria should be glowing because it has that plasmid in it. I also had a black light so I could easily see because sometimes it wasn't as clear the gene expression. I used inoculating loops, had a water bath for the transformation experiment, and many, many plates. So for the first, I had 2 trials where I did this. But I had a trial before that was like a starter run. In that trial run, I had too much lead in my plates, so bacteria wasn't growing the way it should have. Like, there was absolutely no growth and no glowing. So then whenever I came to get these results, I had to take my first results and kind of adjust it so that the bacteria would still be able to grow and I'd still be able to get results. So when I was completing this experiment, I, of course, wore gloves and a lab apron, had my hair tied back, and followed all of the lab safety precautions when dealing with the bacteria. After the experiment and transformation was over, and after I was done with the bacteria, I would wipe everything down with bleach spray and make sure the entire area has been sanitized. And then after I finished the entire project, I made sure that my plates were properly disposed of with autoclaving and that the lead was properly disposed of so it wouldn't be harmful. So you can see here in my first trial of the experiment, this shows the growth of the bacteria plates. I got the most growth on my control plates and my plates with lead pellets. The plates with the water and lead flakes didn't have nearly as much growth. But then by the second trial, you can see that I got a lot more growth on pretty much all the plates except for the lead flakes. Those ones just didn't want to grow bacteria. So then over to my discussion. The results of this experiment showed that the plates with forms of lead in them had less growth and glow than the plates that didn't. So after experimenting, there was virtually no growth on any of the plates with lead in them. The only plates that had any kind of glowing were the control plates, which led me to think that in the presence of lead, the bacteria is more concerned about surviving than an unnecessary form of gene expression. So I needed some kind of statistics to go with this project, so I did the transformation efficiency. So this was strictly on the plates with ampicillin in them. I took the number of colonies from each plate, and then I would put it into the transformation efficiency formula, and it showed that out of all of my plates, I got the highest transformation efficiency on my control plates. So for every milliliter of DNA, I would get 1 to 5 colonies for my control plates. And then I got the least amount of growth on my lead flake plates because for every milliliter of DNA that went in, I would get maybe 1 colony. But most of the time it was 0 colonies per milliliter. So in conclusion, the results of this experiment support my hypothesis and showed that when bacteria is in the presence of lead in any form, the bacterial growth and glowing gene expression will be decreased. And then I would like to thank my BI teacher, Mrs. McMillan, for all of her help, and my mentor, Dr. James West at the College of Worcester.